Sure. By way of background, HER2-positive gastric cancer is an important subset of our disease, and we are focusing on results of KEYNOTE-811, which addressed the unmet need of improving outcomes in those patients in first-line setting in treatment-naive population. So HER2-positive disease occurs in approximately 30% of patients, and gastroesophageal cancer, certainly in the United States, is increasing in incidence. And so KEYNOTE-811 used combination of chemo plus immunotherapy and anti-HER2 drugs to improve outcomes based on the standard. So the TOGA regimen got trastuzumab approved in the United States over a decade ago, but since then, combination of chemo and trastuzumab has been used, but there's no further advances. So KEYNOTE-811 was the first study in a long time to demonstrate improvement in outcomes over the TOGA regimen, over the chemo trastuzumab, and that was in combination of trastuzumab plus anti-PD-1 agent called Pembrolizumab and chemotherapy. We demonstrated first improvement in response rate, and the FDA had approved this combination of Pembro, Traz plus chemo as a conditional approval, just based on first interim analysis. Then we presented the results of the second interim analysis, looking at progression-free survival, which also was improved. And so these papers were published in Nature and in Lancet for reference. And then since then, this most recent final confirmatory approval was based on the final overall survival analysis, looking at combination of chemotherapy with 5-FU platinum plus pembrolizumab and trastuzumab in HER2-positive previously untreated patients. And what we saw and demonstrated is a improvement in overall survival, which was the final analysis, and certainly is important. And we published this data as a letter to New England Journal of Medicine. The median overall survival was 20 months, which, looking at the TOGA regimen, for example, with just chemo, trastuzumab, that survival was less than 15 months. So that's a big leap forward for our patients and improvement in outcome. We also saw that this regimen was very well tolerated. And of course, seeing a regimen that improves response rate, progression-free survival, and overall survival led to the FDA approval. Yeah, so as I mentioned, this regimen was available for HER2-positive disease in United States for quite some time now, based on the additional initial conditional approval. And seeing overall survival data, the FDA confirmed the approval. Of course, if the survival was not as robust, the difference, or there were some, other issues, that approval and the use may have been a question. But I think having the definitive data is a great step forward to confirming what many oncologists do already, because pembrolizumab, trastuzumab with chemo was already on NCCN guidelines, but I think the level of evidence is even more robust now, and I suspect that this is more of a confirmation of what many doctors already agree is the best option for our patients. Well, it's important to understand that these patients, it's not just the median change, the improvement, but the hazard ratio, but also the long-term survivors that make this regimen so promising. And to be able to see plateau on the survival curve and long-term survivors is great to see. I think one important factor is that the FDA recommended that we restrict use of immunotherapy across the board based on PD-L1 status for CPS positive patients, CPS 1 or greater. And to me, I think that one of the more surprising features was that we saw that there was no benefit in PD-L1 low or 0 patients, CPS less than 1. And that was surprising because in our phase 2 data, both in US and then a separate study that was done subsequently in Korea, we didn't see that signal. We saw that patients were benefiting irrespective of PD-L1 status. In fact, our subgroup analysis showed that PD-L1 low patients benefited just as great, if not more. So I was surprised to see that, but it's hard to question KEYNOTE-811 phase 3 data. So for now, the FDA restricts use of chemo, trastuzumab and pembrolizumab in first-line setting by PD-L1 CPS 1 score or greater. I think PD-L1 is important. We should definitely test for it, and plan on acting on it based on your status. But I think HER2 and MSI high really trumps PD-L1. So in terms of the order of the importance of the biomarkers, microsatellite instability is first, HER2 is second, and PD-L1 is third. And I would act on if you have any PD-L1 overexpression and HER2 positivity, and give these patients chemo immunotherapy. Well, certainly this is a chemoimmunotherapy. So if you are able to get immunotherapy, and if you don't have any issues with immune checkpoint blockade toxicity, such as colitis or his predisposition to autoimmune disorders, then you should be able to get these drugs safely. I think it's important to note that most of the side effects we see in these combinations is really from chemotherapy, and the chemotherapy delivery is important. Of course, KEYNOTE-811 used at every 3-week regimen, which is usually capecitabine oxaliplatin. In the United States, we prefer to use FOLFOX, which is every 2-week regimen. And so to mitigate some of the toxicities that we see with chemotherapy, my recommendation typically is to do for our frailer patients, especially and who are maybe elderly and can't swallow pills as well. We do usually FOLFOX with trastuzumab every 2 weeks and pembrolizumab every 6 weeks. I'm glad you asked that question, and there's a lot of interest in my group to understand, are there a subset of patients that we can potentially cure with this regimen? Because a complete response is really seen in it's a double-digit complete response rate, 11%, sometimes higher, and that's in stage 4 disease. So the two main questions that my lab and my collaborators and my group is focused on right now is, one, how do we bring this type of response to the earlier stage disease in perioperative setting? And we're doing that study now in collaboration with MSK, which is the sponsor of the study, and other centers such as MGH and MD Anderson. And two, even within stage 4 disease, but certainly in early stage disease, other patients who we can cure with this regimen. And what we're really developing is a biomarker of combined both circulating tumor DNA and T-cell receptor sequencing to try to develop these type of predictive models.